In this video, let us learn about various connections of the cerebellum and how different nerve fibers form networks within the cerebellum. Although we haven't understood all the functions of cerebellum as yet, cerebellum forms one of those parts of the brain where the connections and internal circuits are very well understood. White matter of cerebellum includes predominantly extrinsic fibers. It includes all the afferent fibers and efferent fibers to and from the cerebellum. They form mainly three fiber bundles which connect the three parts of the brainstem with the cerebellum. The lowest fiber bundle includes predominantly afferent fibers to the cerebellum and few of the efferent fibers from cerebellum. This is the inferior cerebellar peduncle, otherwise known as restiform body. The middle fiber bundle connecting the pons to the cerebellum is made up of entirely afferent fibers to the cerebellum. It's known as middle cerebellar peduncle, or otherwise known as Brachium Pontis. And the upper fiber bundle connecting midbrain with the cerebellum has fewer afferent fibers but mainly efferent fibers from the cerebellum, and this is known as superior cerebellar peduncle, otherwise known as Brachium Conjunctivum. Cerebellum also has some of the intrinsic fibers which include association fibers connecting different parts of the same cerebellar hemisphere. Now this fiber group also includes the fibers which are starting from the cerebellar cortex and projecting to intra-cerebellar nuclei. And then in the cerebellum we have commissural fibers. Unlike the commissural fibers of cerebral cortex, where the commissural fibers connect homotopical points of the two cerebral cortices, in case of cerebellum, commissural fibers are basically crossing fibers. So they can be the afferent fibers or efferent fibers, which will be crossing to the opposite side and then exiting or reaching back to cerebellum. So in truest sense, these commissural fibers have to be grouped with the extrinsic fibers. Now let us first learn about the three major fiber bundles. Let us begin with inferior cerebellar peduncle or restiform body. We have already studied this with medulla. So it is made up of predominantly afferent fibers to the cerebellum. So within the inferior cerebellar peduncle, we have posterior spinocerebellar tract fibers, cuneo-cerebellar tract fibers, and rostral spinocerebellar tract fibers. They will be carrying the proprioceptive and exteroceptive information from the limbs as well as trunk of the same side. Inferior cerebellar peduncle also has vestibulocerebellar fibers, both primary and secondary. Primary fibers are those which are the vestibular nerve fibers, which will be traveling with the inferior cerebellar peduncle to reach the cerebellum directly. Secondary fibers are those wherein the vestibular nerve fibers will have relayed in the vestibular nuclei, and the fibers which begin from the vestibular nuclei, they will form the secondary vestibulocerebellar tract fibers. Now both primary and secondary vestibulocerebellar fibers, they form a surface elevation on the inferior cerebellar peduncle, which is called as juxta-restiform body. Now other afferents within the inferior cerebellar peduncle are the olivocerebellar and parolivocerebellar tract fibers. Now they will start from the contralateral inferior olivary nuclear complex. Now they form the only climbing fiber input to the cerebellum. All other afferents form the mossy fibers, whereas the fibers which are starting from contralateral inferior olivary nuclear complex will form the climbing fiber input. We will study about climbing and mossy fibers a little more in detail in a short while. Then we have trigeminocerebellar fibers and reticulocerebellar fibers, which will also be reaching the cerebellum through the inferior cerebellar peduncle. And within the medulla, we have displaced pontine nuclei known as arcuate nuclei. So fibers starting from the arcuate nuclei will cross over to the opposite side and reach the contralateral inferior cerebellar peduncle. These fibers either travel on the anterior aspect of the medulla as anterior external arcuate fibers, or they travel on the posterior surface of the medulla as stria medullaris. Inferior cerebellar peduncle also contains few efferent fibers from the cerebellum. They include the cerebello-vestibular fiber bundle, which is the bulkier part, or rather which is the major part of the output. And the other output fibers within the inferior cerebellar peduncle include cerebello-olivary fiber output and the cerebello-reticular fibers. Now coming to the middle cerebellar peduncles, otherwise known as Brachium Pontis, they are made up of entirely afferent fibers to the cerebellum. Most of the fibers belong to cortico-pontocerebellar pathway. Few of the fibers may belong to tectocerebellar fibers, as well as the fibers which begin from reticular formation in the pons or some other cranial nerve nuclei in the pons. The surface landmark for the beginning of the middle cerebellar peduncle is the exit point of the trigeminal nerve. So whatever fiber bundle we are seeing lateral to the exit point of the trigeminal nerve, we call it as middle cerebellar peduncles. Now coming to superior cerebellar peduncles, otherwise known as Brachium conjunctivum. This is made up of predominantly efferent fibers from the cerebellum. However, it also has few afferent fibers. One fiber tract, which is the ascending fiber tract within the superior cerebellar peduncle, is the anterior spinocerebellar tract. We know that this tract, when it began in the spinal cord, the fibers cross to the opposite side. It ascends on the contralateral side as ascending spinocerebellar tract, reaches the medulla, traverses through the medulla and pons, reaches the lower end of the midbrain, where the fibers will cross back to the same side either in the superior cerebellar peduncle decussation within the midbrain, or they may reach cerebellum and cross to the opposite side as the commissural fibers. Other afferent fibers within the superior cerebellar peduncles include the tectocerebellar fibers, trigeminocerebellar fibers, and ceruleocerebellar fibers. Trigeminocerebellar fibers here will begin from the mesencephalic nucleus, so they are carrying the proprioceptive information from the head region. Now coming to the efferent fibers from the cerebellum, which are traveling in the superior cerebellar peduncle. In fact, bulk of the efferent fibers from cerebellum will be traveling within the superior cerebellar peduncles. They are classified as uncrossed fibers and crossed fibers. Each group is further divided into ascending fibers and descending fibers. So uncrossed ascending fibers will reach the reticular formation of midbrain, and uncrossed descending fibers will reach the reticular formation of pons and the medulla. Now coming to the crossed fibers, crossed descending fibers will reach the inferior olivary nuclear complex. Crossed ascending fibers will reach the red nucleus. Some of them will reach the red nucleus, of which the fibers which will have started from the interpositus nucleus, that is the nucleus emboliform of the interpositus, will reach the magnocellular part of the red nucleus. So from there, this will be the fibers will be descending as rubrospinal tract. And the fibers which will have started from the dentate nucleus and reaching the red nucleus, they will be reaching the parvocellular part of the red nucleus, and they will project as central tegmental tract to the inferior olivary nuclear complex. And from here, the fibers will be projected back to the cerebellum as climbing fibers. As we have noted earlier, this is the pathway which will be activated whenever there is novelty detection or error detection. So it is an important pathway for motor learning. The second major output which is included in the crossed ascending fiber bundle is to the nucleus, the thalamus, that is both from nucleus interpositus and from the dentate nucleus. The fibers will ascend and cross to the opposite side ventral anterior and ventral lateral nucleus of the thalamus. From the thalamus, the information will be reaching the cerebral cortex. So efferent fibers from the cerebellum which are traveling in the superior cerebellar peduncle are divided as uncrossed and crossed fibers. Both groups have ascending and descending fibers. Uncrossed fibers, whether it is ascending or descending, they will be reaching reticular formation. Crossed fibers, descending ones, will be reaching the inferior olivary nuclear complex. Ascending fibers will reach either the red nucleus or the thalamus. Now coming to, let us summarize the principal inputs to the cerebellum. Cerebellum receives major inputs from vestibular apparatus. That is why one of its important function is maintaining equilibrium. The second major input to the cerebellum is from that is somatosensory input coming from spinal cord and the head region. So basically the individual muscles of trunk and limbs will project through posterior spinocerebellar tract and cuneo-cerebellar tract. Reflex arcs or bigger parts of the limbs will project through anterior spinocerebellar tract and rostral spinocerebellar tract. Similar information from muscles of the face will be projecting through trigeminocerebellar input. They will all be projecting either from the spinal cord or from the trigeminal nuclei to the cerebellum of the same side. The third input will be visual and auditory inputs coming from the tectum. So all these three inputs, that is vestibular input, input from the spinal cord and trigeminal input, and visual and auditory input from tectum, they will all be ipsilateral inputs. Cerebellum also receives contralateral input from the cerebral cortex via the pontine nuclei, that is cortico-pontocerebellar pathway, and it will also receive contralateral inputs from the inferior olivary nuclear complex. As we know, all other inputs to the cerebellum form the mossy fiber inputs, whereas the fibers coming from the inferior olivary nuclear complex form the climbing fiber afferents. Now coming to the principal outputs from the cerebellum. Cerebellum, though it has maximum number of neurons in the entire brain, in fact it has more than 50% of neurons within its cortex, the cerebellar cortex does not project to any major structure outside the cerebellum. Instead, they will all be projecting to the intracerebellar nuclei. Very few fibers from the cerebellar cortex will project outside the cerebellum. They will be reaching only the vestibular nuclei. Now, from the intracerebellar nuclei, the projection is to vestibular nuclei and to the reticular formation. Both these sources will be receiving bilateral projection from the cerebellar cortex. Intracerebellar nuclei will also project to the contralateral inferior olivary nuclear complex, contralateral red nucleus, and contralateral ventral anterior and ventral lateral nucleus of the thalamus, from where it will be sending information to cerebral cortex. So these are the principal output areas from the cerebellum. If you notice these principal areas of output, all these will in turn be projecting to spinal cord so as to control the motor activity. So we have vestibulospinal tract, reticulospinal tract, olivospinal tract, rubrospinal tract, and finally corticospinal tract. Let us now study the cerebellar circuitry. What we have here are the layers of grey and white matter. So the outermost layer in the grey matter or the cortex of the cerebellum is the molecular layer, where we are finding the dendrites of the Purkinje cells. The middle layer consists of a single layer of cells that is known as Purkinje cell layer, and the deepest layer of the cortex is the granular layer. So molecular layer is the superficial layer in the cortex. Then we have Purkinje cell layer, which contains a single layer of Purkinje cells, and then the deeper layer is the granular layer. Deep to the cortex we have white matter. Within the white matter we have intracerebellar nuclei. Now afferents to the cerebellum include two types of fibers basically, the mossy fibers and the climbing fibers. Now mossy fibers, as they reach the cerebellum, they are coming from all the parts of the CNS except from the inferior olivary nuclear complex. So as these fibers enter the cerebellum, they send a collateral to the intracerebellar nuclei, which will be excitatory input. And then each mossy fiber divides into 50 to 100 branches. Each of this branch will synapse with dendrites of a single granule cell. Now axons of these granule cells will reach the molecular layer, where they bifurcate into two branches. Now these branches will run in the opposite direction as parallel fibers, synapsing with 500 to 1000 Purkinje cells in a row on either side. So one mossy fiber first had divided into 50 to 100 branches. Then each branch will synapse with one granule cell, which will then divide into two branches. Each branch will be synapsing with 500 to 1000 Purkinje cells. So a single mossy fiber is capable of exciting about 100,000 Purkinje cells. With each Purkinje cell, the parallel fibers will make only single synapse. I'll repeat this. With each Purkinje cell dendrites, the parallel fibers are these axons of the granule cells, they will make only a single synapse. So their excitation may not be sufficient to start an action potential. So it needs to be strengthened by input coming from multiple parallel fibers. So each Purkinje cell actually synapses with close to 2 lakh parallel fibers. So when the input is strong enough, it will result in an action potential. The second type of afferents to the cerebellum include the climbing fibers. They are named as climbing fibers because they climb straight to the molecular layer. Again, these are the fibers which are coming from contralateral inferior olivary nuclear complex, and only that. And again, when they enter the cerebellar cortex, they send collateral branches to the intracerebellar nuclei, just like mossy fibers, which are excitatory. Once they reach the molecular layer, they synapse with dendritic tree of one single Purkinje cell. So each climbing fiber synapses with dendritic tree of one single Purkinje cell. However, each climbing fiber makes hundreds of synapses with the dendritic tree of the Purkinje cell. So parallel fibers would synapse with many cells, but with each cell it makes only one single synapse. Here the climbing fiber synapses with only one cell but makes multiple hundreds of synapses. Okay, so that's the difference between the mossy fiber ending and climbing fiber ending on the Purkinje cell dendritic tree. Now, apart from this granule cells and Purkinje cells, cerebellar cortex has few more types of neurons. Within the molecular layer, very close to the surface, we have what is known as stellate cells. The dendrites of these cells are also excited by the parallel fibers. The axons will go and inhibit the trunk of the dendritic tree of Purkinje cells. This is a feed-forward inhibition. There is another set of cells within the molecular layer which are located at a much deeper plane. They are called as basket cells. Again, dendrites of these are excited by the parallel fibers. The axons will run and synapse with close to 200 to 250 Purkinje cells, and around the Purkinje cells they will form a basket and they will synapse with initial segment of the axon of the Purkinje cell, again inhibiting the Purkinje cells. This is again a feed-forward inhibition. And then we have a fifth set of neurons within the granular layer. This is known as Golgi cell, the dendrites of which are again excited by the parallel fibers. Axons will, however, end in the dendrites, making a inhibitory synapse with the dendrites of granule cells. So stellate cells and basket cells are found in the molecular layer. Golgi cells and granule cells are found in the granular layer, and Purkinje cells are found in the Purkinje cell layer. So these are the 5 types of neurons within the cerebellar cortex. If you notice all these fiber network whichever we have mentioned so far, granule cells, Golgi cells, stellate cells and basket cells, all these neurons, especially the granule cells which are the majority in number, all of their axons are ending within the cortex. The only cerebellar output comes as the axons of Purkinje cells. Again, these cells will not project out of the cerebellar cortex. Most of these cells will project only to the intra-cerebellar nuclei, and their projection is inhibitory. So although cerebellar cortex has got maximum number of neurons, the final story, bottom line, is their only projection, which is the axons of the Purkinje cells, is inhibitory output. However, few Purkinje cells may exit from the cerebellum. They will reach only the vestibular nuclei. Purkinje cell axons will also send collaterals, which will be reaching the axons of the Golgi cells, or they may reach the dendritic tree of the surrounding Purkinje cells. So main output from the cerebellum is only from the intracerebellar nuclei, that is vestigial nucleus, nucleus interpositus, which includes globose as well as emboliform, and the dentate nucleus. So the neurons of these nuclei alone will send the efferent fibers outside. Very few Purkinje cell axons will leave the cerebellum to reach the vestibular nuclei. So coming to some of the interesting features here, we have what is known as synaptic glomeruli. Each mossy fiber, when it ends as a bulbous dilatation that is known as a mossy fiber rosette. It synapses with the dendrites of granule cells, and these dendrites of the granule cells will be receiving inhibitory input from the axons of the Golgi cell. So this area where these three types of fibers are meeting, it's known as a synaptic glomeruli. They are found in the granular layer of the cerebellar cortex. Now, second interesting feature is each parallel fiber is synapsing with 500 to 1000 Purkinje cells on either side in a row. So obviously the first Purkinje cell will get excited much earlier than the Purkinje cell number 400 or 450 or 500 or even 1000. So there is a time gap between the excitation of adjacent Purkinje cells. Probably that is one of the way in which cerebellum controls sequence of the movements. The third one is, although cerebellar cortex has the maximum number of neurons in the entire brain, the Purkinje cells form the only output neurons, and their output is inhibitory. Then we have feed-forward inhibition done by both stellate cells and basket cells. They will allow only the central microzone of active Purkinje cells. The cells of Purkinje cells on either side will be inhibited by these stellate and the basket cells, known as lateral inhibition. So this will result in neural sharpening. So only that central microzone will be excited, the surrounding cells will be inhibited. That's resulting in neural sharpening. There is also feedback inhibition, which is done by Golgi cells, which will be inhibiting the granule cells, or it will also be done by the collaterals of the Purkinje cell axons themselves. Now this is on-beam inhibition, that is at a local point there will be inhibition. So both these feed-forward and feedback inhibition will be seen in case of cerebellum, which will result in exciting only the required number of Purkinje cells in the selected microzone. Now coming to comparing the mossy fiber with the climbing fibers. So mossy fiber include all the inputs except olivocerebellar, and climbing fibers include only the olivocerebellar afferents. Mossy fibers synapse with 100,000 of Purkinje cells, whereas each climbing fiber synapses with a single Purkinje cell. Mossy fiber makes a single synapse with one Purkinje cell, whereas climbing fiber makes multiple synapses with one Purkinje cell. The excitation caused by the mossy fiber results in a simple spike within the Purkinje cell, whereas excitation caused by the climbing fiber will result in a complex spike. That is what will result in corrective behavior, or as I said, this whole pathway gets excited because of novelty detection or error detection. Because of this complex spike, it will result in corrective behavior or the motor learning activity. Now coming to the efferents from intracerebellar nuclei. Those which are starting from the vermal zone or the flocculonodular zone, they will receive input from posterior spinocerebellar tract, cuneo-cerebellar tract, but mainly from vestibulocerebellar tract, olivocerebellar tract and the reticulocerebellar tract. They will be projecting to vestigial nucleus. Few of the fiber output will also be directly the axons of the Purkinje cells itself. And from these vestigial nucleus, the output is mainly to bilateral vestibular nucleus and also to the reticular formation, from where the output will reach to the spinal cord as vestibulospinal tract or the reticulospinal tract. From the paravermal zone, which is marked in blue here, again these paravermal zones will be receiving inputs from the posterior spinocerebellar tract, cuneo-cerebellar tract, pontocerebellar tract fibers, which are starting from the M1 region, that is primary motor area, Also, it will be receiving inputs from the olivocerebellar and reticulocerebellar tract. Olivocerebellar and reticulocerebellar tract fibers will be reaching all parts of cerebellum. Now, the paravermal zone will be projecting to nucleus interpositus, and from the nucleus interpositus, the output is either to the contralateral magnocellular part of the red nucleus or to the contralateral ventral anterior and ventral lateral nucleus of the thalamus. From the red nucleus, the fibers will be projected to spinal cord as rubrospinal tract, and from the thalamus, the input will be to the cerebral cortex, predominantly to the primary motor area. Now, rest of the lateral part of the lateral zone of the hemispheres, they will be receiving corticopontocerebellar tract fibers, that is pontocerebellar tract fibers, also olivocerebellar and reticulocerebellar tract fibers. Now, from this lateral part of the lateral zone of the cerebellar hemispheres, the Purkinje cells will project to dentate nucleus. From the dentate nucleus, the efferents will travel to the contralateral parvocellular part of the red nucleus. From there, it will reach the inferior olivary nucleus via the central tegmental tract. Now, this is the part which is involved in motor learning. Or they will reach the ventral anterior and ventral lateral nucleus of the thalamus, and from there to the premotor area and supplementary motor area, which is involved in motor planning. So, you are seeing that the vermal zone and the flocculonodular lobe predominantly receives vestibulocerebellar tract input plus few fibers from the spinocerebellar part, and they will be projecting mainly to the fastigial nucleus and from there to the vestibular nuclei and reticular formation. The paravermal zone receives mainly the spinal cord inputs, and they will be projecting to nucleus interpositus, and from the nucleus interpositus they reach the red nucleus and then to the spinal cord via rubrospinal tract, or from there they will project to the contralateral thalamus and to the primary motor cortex. The lateral zones, which are involved in motor learning as well as higher functions, they will be projecting to the dentate nucleus. They will receive the input from the cerebral cortex as pontocerebellar tract fibers, and they will project to the dentate nucleus. From the dentate nucleus, the input will be, the output will be to the parvocellular part of the red nucleus, which is then going to project to the inferior olivary nuclear complex, or it will project to contralateral thalamus, ventral anterior and ventral lateral nucleus. From there, it will reach the premotor area and supplementary motor area. So, rounding up the vestibulocerebellar function, this is basically involved in balancing. This has a bilateral control. So, vestibular apparatus will be sending its output through vestibular nerve, which will project to the vestibular nuclei. Both vestibular nerve and the vestibular nuclei will project to flocculonodular lobe and the vermis part of the cerebellum. From the flocculonodular lobe and vermis, the projection is to the fastigial nucleus. From the fastigial nucleus, as well as some of the Purkinje cells in the flocculonodular lobe and vermis, will project back to the vestibular nuclei. Now, this projection is bilateral. That is how the posture is maintained, because it's going to project predominantly to the trunk muscles as well as to the proximal limb muscles thereafter. From the vestibular nucleus, the output is to the spinal cord through vestibulospinal tract, which will be facilitator for the extensor muscles, therefore maintaining the posture, and also to the third, fourth and sixth cranial nerve nuclei via the medial longitudinal fasciculus, which will be involved in maintaining the conjugate eye movements, which is again important for maintaining the vestibulo-ocular reflexes. And fastigial nucleus will also project to reticular formation, which will again then send the output to the spinal cord through reticulospinal tract. Now, coming to the spino and neocerebellar part of the cerebellum, they will be involved in controlling the limbs, distal limbs, motor learning and planning. Now, they have ipsilateral control, so they receive information from the same-sided tracts, and they will be projecting to the same-sided areas. So, spinal cord as well as the fifth cranial nerve nuclei will be projecting via the posterior spinocerebellar tract, anterior spinocerebellar tract, cuneo-cerebellar tract, or rostral spinocerebellar tract. And from the trigeminal nucleus, it will be projected as trigeminocerebellar tract. The neocerebellar area will receive predominantly input from the cerebral cortex as corticopontocerebellar tract fibers. Now, both these areas, that is spinal cord as well as cerebral cortex, will be projecting to cerebellar hemispheres, that is both intermediate zone and the lateral zone. From the cerebellar hemispheres, the projection is to nucleus interpositus and dentate nuclei. From these interpositus and dentate nuclei, the output is to the reticular formation, to the contralateral inferior olivary nucleus, contralateral red nucleus, and contralateral thalamus. Thalamus then projects to the cerebral cortex. Red nucleus, especially the parvocellular part, will be projecting to the inferior olivary nucleus through central tegmental tract, which will then send input back to the cerebellar hemisphere through olivocerebellar tract fibers, that is the climbing fibers, which we have done earlier. Now, if you notice these outputs from the reticular formation as reticulospinal tract, from inferior olivary nucleus as olivospinal tract, red nucleus as rubrospinal tract, and cerebral cortex as corticospinal tract, the fibers will be projected back to the spinal cord, which will help in controlling the movement of the muscles which are controlled from spinal cord. So that is how spinal cord has control on the movements. Directly it does not initiate movement, but it will send input to all the motor activity controlling centers, which will then send the input directly to the anterior horn cells in the spinal cord. This was a little lengthy video, I understand, but we wanted to do this in a single setting so as to have a better understanding about connections of the cerebellum. So go through this once more to understand the cerebellar circuitry as well as all the various connections to the cerebellum. Thank you very much. Hope you enjoyed this video. Thank you for patient listening. You can visit the site for other videos on neuroanatomy.